As we saw, proteins are hydrolyzed in the stomach and the small intestine through a class of enzymes called proteases in the presence of water to make our amino acids. At this point, the body has to take stock. What do we need from these amino acids? We can use these amino acids and undergo anabolic processes to build up proteins to serve the function. These amino acids can also undergo catabolic processes and provide energy via ATP to our cells. However, this is only going to happen when our carbohydrate and lipid supplies are low. So proteins are not our primary source of ATP. But if we are like in a starvation mode, then the body is going to start to catabolize our amino acids. That's the pathway we are going to follow now. I'm going to use alanine as an example so that you can see how this catabolic process occurs. Now, when we look at the citric acid cycle, because that is where our high energy electron carriers are made for the electron transport system, the electron transport chain that then can undergo oxidative phosphorylation and make our ATP. So many words. All the compounds in that citric acid cycle have only carbon, hydrogen, and oxygen. Amino acids also have a nitrogen. So we need to remove that amino group first. We're going to do this through a process called transamination. Trans means to transfer, and amine. So this is a transferase enzyme that is going to move a functional group. Here we have alpha ketoglutarate. So alpha ketoglutarate is a derivative of glutamic acid or glutamate. Here's the R group. We're not really paying attention to that R group because it's not the reactive side of this compound. Here we have our alpha keto. So a ketone on the alpha carbon that's next to the carboxylic acid. So that is where the alpha ketoglutarate comes from. We are going to move that amine onto that alpha ketoglutarate. That R group remained the same. Like I said, we're not going to really pay attention to that. But it is the keto group that gets reacted with. So we remove that ketone, it gets transferred. We'll get to that in a minute. We're going to remove that ketone and put the amine group onto that alpha ketoglutarate. So we are going to take this amine with the hydrogen from the alpha carbon of this alanine, and that is what's coming onto the alpha carbon of the glutamate. I'm not going to ask you specifics. What I'd like you to be able to see are where the changes are occurring and what is moving between each of these structures. So now that we've transferred that amine group, what happens to the carbon backbone that was our amino acid? So we have this carboxylate group, that alpha carbon, and the R group. Well, that is going to create what's called an alpha keto acid. So this ketone that was part of this alpha ketoglutarate essentially is getting shifted or transferred onto that alanine molecule. So here we have two transfers occurring. We have a transfer of the ketone from the alpha ketoglutarate to alanine. We have the amine group from the alanine going to the glutamate. So here we just have transferings of these functional groups because now we have just carbon, oxygens, and hydrogens that can then move forward into our citric acid cycle. We'll talk about that at another time. Really, what we want to know is what happens to this amine. Where's it going, right? So tracking this amine group, because we cannot incorporate it, we are breaking this down. We are going to undergo oxidative deamination. D, the removal of and amine via an oxidation reaction, which means that this alpha carbon that had the amine group is going to lose that amine, and it's going to get oxidized back into alpha keto acid. What gets released is ammonium. Ammonium. We cannot use ammonium in our body, therefore this is going to undergo what's called the urea cycle, and then that urea will move to the kidney to then be flushed out as urea. So urea is the final compound that is made for our nitrogens to be then excreted from our body. So to give you an idea, this is what urea looks like. So it's a high concentration of nitrogen in this very small compound that then we have to excrete. So that is the final resting place of our nitrogen. It does leave the body if we are catabolizing our amino acids down for energy.